Assalamualaikum and hi everyone! First of all, I would like to greet our respected lecturer, Dr. Siti Hajar, and all of you watching this video. My name is Shalia and I'll be the first presenter for our group presentation. The title of our presentation is Application of Volumetric Analysis, Determining Salt Content Sodium Chloride in Food. But first, let's get to know the team.
Zleka, Sheila, Stefan,
Shalia.
Now, let's begin with the introduction. So, what exactly is volumetric analysis? Volumetric analysis is a method in chemistry that helps to find out how much of a substance is present in a solution. It is a type of quantitative analysis, which means it gives us exact amounts or concentrations. This technique usually involves a process called titration, where a solution with a known concentration is slowly added to another solution until the chemical reaction is complete. Titration is commonly used because it is simple, accurate, and does not require expensive equipment. It uses tools like burettes, pipettes, and volumetric flasks to measure liquids precisely. Volumetric analysis is very useful in many fields such as food testing, medicine, and environmental science because it helps scientists and industry make sure their products are safe and meet quality standards. The common use of volumetric analysis is in the food industry to measure the sodium chloride content in food products. Knowing how much salt is present, it is important for food quality, taste, and safety, as well as to follow health guidelines. Titration is a simple, accurate, yet affordable way to check the amount of sodium chloride in different foods, making it very useful in food testing labs. So now let's move on to the next part of our presentation.
I am Zleka and I will explain the methodology used in this experiment. We use two techniques which are titration and isothermal titration calorimetry. For titration, it is a process where titrant is slowly added to a sample until the chemical reaction is complete. This point is called equivalence point. It is usually detected using a color change or pH change. Titration is commonly used for testing acids, alkalis, and even sugar levels. Meanwhile, ITC is used to measure the heat change during titration and helps us understand how molecules like proteins or even enzymes interact. Next, these are the materials and apparatus used. First, we use food sample like soup or snacks, silver nitrate solution, potassium chromate as indicator, distilled water, pipette, burette, conical flask, and other basic tools. Now, let me explain the procedure. About 5 to 10 grams of food is crushed and mixed with distilled water. The mixture then filtered to get a clear sample. 25 ml of the sample is transferred into a conical flask. Meanwhile, potassium chromate is added, which gives a yellow color to the sample. The burette is filled with silver nitrate. Then, we titrate slowly while shaking the conical flask. The end point is when the color changes from yellow to brick red. This means silver chromate is formed. Finally, we record the volume of silver nitrate used. This is the chemical reaction that occurs. Silver nitrate reacts with sodium chloride in the food to form silver chloride and sodium nitrate. A white precipitate forms first. When all chloride ions are used up, excess silver ions react with the indicator potassium chromate, forming a brick red precipitate silver chromate, which marks the endpoint. For calculation, we use the formula molarity times volume to find the mole of silver nitrate. Then we use stoichiometry, which is 1 mole of sodium chloride equals 1 mole of silver nitrate. From there, we calculate the weight of sodium chloride and the percentage of salt in the food sample. For the theory behind this experiment, titration is used to determine how much of a substance is present in a solution. We add a titrant with a known concentration to the analyte until the reaction is complete. This is the equivalence point. Indicators of pH meters help detect the point. There are different types of titration, such as acid-base titration, precipitation titration, complexometry, and redox titration. In our experiment, we use precipitation titration.
My name is Akila Hazimi. Today we're gonna look on example of results. Here we have simulated example of the result for salt content in different food samples. In this experiment, 3 food items were analyzed, which is tomato soup, Mr. Potato chips, and a cupcake. For each sample, a mass of 5 grams was used. The amount of 7 nitrate solutions added varied depending on the sample. 12.5 ml for the tomato soup, 20.0 ml for Mr. Potato, and 8.0 ml for the cupcake. Using the titration formula shown below the table, we calculated the mass of salt NaCl in each sample. The salt content percentage was then defined using this formula. NaCl percent equals to mass of NaCl divide mass of food sample and then multiples 100%. As shown in the result, tomato soup had a salt content of 1.46%. Mr. Potato had highest salt content at 2.34%. And then the cupcake had lowest at just 0.94%. The bar graph on the right visualizes these values, making it easier to compare the salt contents between the different food. It's important to note that this table is only simulation and is not based on an actual experiment.
Hello everyone, I'm Shihan, and now I'll be sharing the conclusion for our topic. So, to wrap up our presentation, we've learned how volumetric analysis, especially titration, is really useful when it comes to figuring out how much salt or sodium chloride is in food. In our experiment, like Zulaika already said in the methodology part, we use silver nitrate as the titrant and potassium chromate as the indicator. When the chloride ions react in the food sample, a brick red color appears, so we know that the titration is done. This helps us calculate the exact amount of NaCl in the food. This method is super important for the food companies because it helps them keep their product consistent and safe to eat. It is also make sure they follow health rules because eating too much salt can lead to health problems like high blood pressure. So in conclusion, volumetric analysis is the simple and best way to help make sure the food we buy is not only tasty but safe and meets the right standard.